So there are two things I want to do right now. One is I want to briefly finish our discussion of methods for phylogenetic trees, and then I'm going to go through a series of sort of questions that can be addressed using molecular phylogenetics, with the hope being that this will sort of help prompt your thinking too, at least to some extent, about the types of questions you might be able to think about with the analytical exercise that you're starting. So to begin, I want to just briefly finish up talking about methods for phylogenetic inference. And really the last thing I want to say about this is we talked last time about optimizing branch lengths in trees. We sort of talked about how there's a series of steps that are taken to find with a computer algorithm to find the best topology to optimize the parameters for the nucleotide substitution matrix and to optimize the branch lengths, all through maximum likelihood is usually the criteria used, and that's again what you're going to be doing. The last thing I want to say is that from that you'll get a tree with some series of branches and branch lengths. Well, how confident should you be in that tree relative to alternative trees? So I want to think about the question of how confident one should be in a given tree relative to others. So how confident can we be in our best tree relative to other trees? And the way to do this in particular is that we can think about how confident we are in individual branches or clades, or sort of bifurcations or clades on the tree. There's a number of approaches that are used for this. This includes looking at something called bootstrap support, which usually ranges between 0 and 100%. And bootstrap support is associated mostly with parsimony-cladistic analysis, and it's essentially a measure of how often you would generate that bifurcation or clade in the tree if you subsampled the data set. A more common, currently, and more intuitive, I would argue, way of assessing support is something called Bayesian posterior probabilities. And I have an example I have an example of this over here. So here you're looking at a tree of Plasmodium. This includes the vector that causes malaria. It's responsible for malaria. And you're looking at the tree reconstructed of Plasmodium in humans and some other primates. The thing I want you to see, so here we have a bunch of branches. Here's our best topology. You'll see all these numbers on here, 1.0, 1.0, 0.91, 0.68. That is from our computational algorithm, and this is done in a Bayesian framework, which is somewhat related to likelihood, is simply the probability that is in fact the appropriate branching pattern. So essentially there's a probability of 1 that P. Gondaria and this Plasmodium spa are each other's most close relatives. Similar, here we have P. annui from Taiwan and from some leaf monkey. Again, a posterior probability of 1 that's the true branch. Notice that some are lower. For example, we're slightly less confident. The probability is only 0.91 of this split here between these two Plasmodium species and essentially all these other ones. And here we have even less confidence, 0.68 for this bifurcation right here. That says, okay, we split into a group that includes Semioval and Fialdi versus all these other guys here. Those are posterior probabilities. The last thing you'll see occasionally are using likelihood ratio support. Oh, let me go, just to be clear, this ranges from values of 0 to 1. The last one you'll see is likelihood ratio support. This is actually what you're going to use for your analytical exercise. It also ranges from 0 to 1. And it's similar to a Bayesian posterior probability in the sense that it's a measure of the relative likelihood of a tree with versus without that bifurcation. And 0 means that tree with the bifurcation is equally probable as a tree without the bifurcation. 1 means the tree with the bifurcation is much more probable, or the data rather are much more likely given that tree than the alternative tree. So similarly, scaling between 0 and 1. So this can tell you which individual branches on a tree you should be confident is we probably have this right, versus other ones where we don't really know, and there are alternative solutions to this problem that would give us a similar good answer. So that's the last thing I really want to say about constructing trees. Now I want to talk some about what questions we can address with trees. So let's think about using phylogenetic phylogenetic trees to answer questions and test hypotheses. This is really why we want to construct them, right? To sort of answer questions. Now, the first thing I'll think about is in some ways maybe the most trivial of these, in the sense that it's maybe the most obvious thing you do with a phylogenetic tree, and that is to get at questions related to systematics. In other words, taxonomic classification of organisms. And the basic idea here is that taxonomy should reflect evolutionary history. In other words, and the what we want in systematics is we want to group individuals into genera, families, and other higher level groups based on their shared evolutionary history, such that these groups that we're identifying are monophyletic groups. And just in case, so just in case you're not familiar with that, by a monophyletic group, this is a group or clade, just in essence another word for group in this context, to consist of an ancestral species and all of its descendants. Let's think about an example over here real quick. So here's my beautiful tree. Whoop. Whoop. All right, and once again, we're going to pop some species on here. We're going to have species A, species B, species C, D, and E. So monophyletic group is a group that contains the ancestor of that group and all of its descendants. For example, the group that includes A, B, and their ancestor here would be a monophyletic group. So let's think about some of these. So monophyletic groups include the group that is A, B, and C is one monophyletic group, A, B, and C. So is AB. AB is also a monophyletic group. So AB and its ancestor would be monophyletic, ABC and its ancestor is monophyletic, as is DE and its ancestor. This contrasts with other potentials, such as a paraphyletic group, which is not what we're really trying to create when we're doing phylogenetics, or when we're basing systematics or taxonomy on phylogenetics. A paraphyletic group includes the ancestor to a group and some of its descendants. For example, BC would be a paraphyletic group in the sense that you would imagine a group that includes so this ancestor to A, B, and C, this ancestor to A and B and C, but not A. Be a paraphyletic group, as would sort of similarly AC. The sort of worst case scenario, what we're really trying not to have our systematics or taxonomy reflect, are polyphyletic groups. These are groups that do not include in essence, the ancestor to the group in question even. So a good example of this would be BD or CE. Right? B and D or C and E were sort of would both be considered polyphyletic groups. So we're really, the key here again is that we're aiming for monophyletic groups. So we need a tree to identify monophyletic groups to be able to base our systematics and taxonomy on this. Another reason why we might be interested in phylogenetics for systematics is that phylogenetics can reveal hidden species diversity. All right, so let's look at this for a second. Here's an interesting example from a few years ago with this Straptes fulgurator complex. Now, this is a skipper, which is closely related to a butterfly. It is the adults of what I'll argue in a second could be considered maybe 10 different species. look identical. So if you look at the adults in the field, that you see one thing, it looks like one species phenotypically. However, some individuals went and did a bunch of DNA sequencing and constructed a phylogeny of this of individuals, essentially. In this case, this is really which is a bit odd, but a phylogeny of individuals for many of these skippers. And here's the phylogeny that we see. And the sort of point I want you to get from this is that there appear to be sort of various groups of individuals that are fairly closely related to each other and more distant related to other things. So in other words, it seems like there are sort of meaningful clades within this group. Well, it turns out that if you rear the larvae from these different, and these are separated by these blue lines, right? So here's one of them, trigo, celt, here's another one, numpt, here's another one. It turns out if you rear larvae belonging to these different, what are essentially genetic groups here, the larvae actually look different. So at the level, at the caterpillar stage, these all look very different. It's just the adults look the same. So they had a single name because people based it on the adults. They saw the phylogenetic structure within the tree and then went back and looked more closely at the organisms and said, oh wow, we do actually have what in the end I think is 10 different things here. So here's a case where constructing the phylogeny from molecular data led researchers to hypothesize about different taxonomic unit species lineages within that group that were then verified to some extent by the morphology of the larvae, that these really do correspond to something meaningful. So that's one reason why you might be interested in molecular phylogenetics and constructing trees. Another reason would be for dating historical, way over there, oops, okay. For dating historical speciation events. Trying to get at when speciation events happened. All right, so the basic idea here is that we can use branch lengths to infer the divergence times of different species on a tree. And specifically, under neutral theory, right, meaning that under a model where evolution is dictated mostly by, at least allelic substitutions occur primarily through mutation and fixation by genetic drift, with again positive selection contributing little to evolution. Under neutral theory, the expected rate of substitutions per generation is simply equal to mu, where mu is the mutation rate. So the mutation rate is proportional to the mutation rate gives you the expected number of substitutions per generation. Specifically, right, so if that meaning that if we let k equal the number of substitutions between a pair of species, then k is equal to mu times t, where here t is the number of generations separating those species. So again, let's look at a little example here. Here's another quick phylogeny. Again, we'll sort of just put 3 species on here, A, B, C. So if I know the number of substitutions, which again are proportional to the branch lengths between these species, then I have k, right? And if I want to know how long ago we have divergence happen, well, if again k is equal to mu times t, then sort of by definition t is equal to k divided by mu. So if I take the number of substitutions and divide by the mutation rate, I'll have the divergence time. So as an example, say I have a pair of species that differ by 10 substitutions total. And let's say that's out of, so there's 10 substitutions, right, out of 1,000 base pairs, right? Then the substitution rate per base pair is simply 10 over 1,000, or 0.01 is k. Now let's say the mutation rate per base pair, say mu is something like 1 times, let's say 1 e to the minus 7 is what we'll take as our mutation rate. Well, then all I would do is 0.01 divided by 1 e to the minus 7th, which if we pull up a trusty little calculator real quick, that would be 0.01 divided by 1 e to the minus 7th, gives us 1 to the 5th, 1 e to the 5th. Is the number of generations that have separated these two taxa. I'll point out though that the divergence time between them is not 1 e to the 5th, really, but it's half of that, because 1 e to the 5th is the total time that's elapsed since they've been separated. So in other words, it's this time plus this time, from B back to the ancestor, A back to the ancestor. So the actual time since divergence is 1 e to the 5th divided by 2. So you could use that to get an estimate of how long ago a pair of species split based on the nucleotide differences between them in a phylogenetic context. Now, this is a cool idea, right? It'd be really neat to be able to date historical events like this. But I want to point out that, so then this sort of idea is often referred to as a molecular clock, right? So the molecular clock is a cool idea. And there's some caveats that have to be taken into account, though. So, but caveats exist. First, to recognize this is a stochastic clock. In other words, if I say that on average you get a substitution at a base pair, at any base pair, say in a million years, you get one substitution on average per million years. That doesn't mean that every million years you get a substitution, right? That's just an average. This is a stochastic process. So there's going to be a bunch of noise in this molecular clock. So it's a stochastic clock. So it's not giving us an exact answer. The other thing that we should be aware of is that we need to know the mutation rate, mu, right? Well, mutation rates can vary among species and among genes, right? So this is going to complicate things, making sure we have a reasonable mutation rate, and it's often is very difficult to get at unless you can do studies to look at sort of actual mutation rates in organisms by looking at, say, sequencing of sperm from individual males, things of that nature, which is very costly. You can partially get around this, partially overcome this problem with fossils for tree calibration. The basic idea here is that if you have a tree that's perhaps more complicated than the one I have here, but let's just take this one for example. Say you have a fossil that represents an organism that existed roughly right before the split between C and AB. And you can date that fossil using fossil dating methods that are not molecular methods. And you can say, okay, I have a fossil that puts this split at 10 million years ago. You can use that then and basically say, okay, if I know that one, I have T for this split, and I know how many substitutions I have between C and A or B, I can then estimate mu. So treat mu as the unknown instead of T. I can then use that mu to infer the divergence time for other pairs of species where I don't have a fossil. In other words, you calibrate a few points on the phylogeny based on the fossils and then use that to get an estimate of mu that you can apply to the rest of the tree. This can be a fairly powerful approach, assuming mutation rates don't vary substantially over the group of species and assuming you have enough fossils that you can date more accurately and place appropriately on the tree. All right, so what else can we do with trees? Another thing we can do with trees is we can use them for what's called ancestral character state reconstruction. The idea here is we're trying to infer the ancestral state. In other words, back at points in times of various characters, not necessarily the ones we use to construct the tree. So say we've constructed a tree like this one here based on DNA sequence data, we can then say, okay, species A and B are both hairy species, C isn't. Can we infer what the status of hairedness was back at the ancestor here between A and B or back at the root of the entire tree? There's sort of two general approaches to this. One uses the principle of parsimony. This is again this idea that the simplest solution is the best solution. In other words, that the ancestral character state reconstruction that is the best is the one that requires the fewest changes along the tree. And this was the sort of the only way this was done for quite some time. There's a couple of problems with this. One, similar to before, is that it's not always the case that the simplest solution is in fact the best solution. Also, if you have selection acting, sometimes favoring a trait, sometimes selecting against that trait, this could give you a woefully wrong answer. There's a more popular approach now that is better and partially overcomes these problems. That actually tries to is model based. So an alternative. We have alternative model based approaches that estimate character transition matrix and use this to infer the probability of changes in the character at different points in the tree. This is usually done in a probabilistic fashion. So, and I'll sort of just to clarify maybe a little what I mean here is so we have this character transition matrix. So picture a little matrix here. And we'll make it. Imagine we have two character states, state A and state B. Different colors, different life histories, however you want to sort of conceptualize these. And we have character A, character B, character A, character B. So you're trying to estimate this matrix that describes the probability of, if you're currently in state A, sort of staying in state A, right? So you could be in state A at some point. So imagine starting from the root, or actually it makes more sense for this. Imagine starting from species A. I'm in state A. I have blue color. I can then say, okay, what's the probability going back along this branch of staying in state A? That would be this element of the matrix, or the probability of going from state A to state B as I go back in time. You could also then have the probability of going from state B to state A. So in other words, if species A is in state A and species B is in state B, they could each stay in the respective state up to some point back in time, but at some point one of those has to converge to the other one. So B either switching back to A or A back to B. Of course, the last thing on here would be B staying in B. So the idea is that you're estimating these transition probabilities from the data and using this in a probabilistic sense to estimate the probability of different character states at different points back in time. This is something you'll do with the first part of the analytical exercise, and something you could potentially do. So I'm sorry, you'll do it there not in a formal manner, though. You'll just do it in a qualitative manner, taking the idea basically of parsimony. But you could do this in a you could do this in either a simple sense or a more complicated sense as part of your question if you wanted to for the analytical exercise. And I'll give you an example of what this sort of looks like. The example I want to think about for character state reconstruction involves toads and snakes. So toads produce bufadinolides. And these are toxic to predators. They can cause essentially cardiac arrest. It turns out that some snakes have specialized on toads anyway. These snakes can detoxify this compound. Now, so you have snakes that specialize in toads, can detoxify the bufadinolides. Then it looks like there are two amino acid substitutions in the sodium-potassium pump that are responsible for resistance, at least partially responsible for resistance. This is a little more complicated, but I don't want to worry about the details. So we can score the presence of these amino acids, in other words, which is essentially a way of scoring the ability to feed on these toads, and then reconstruct the ancestral character states back in time for that. And that's, yeah, my picture went away. Let me fix this. All right, there we go. That's better. So what you're looking at here is a phylogeny of snakes. Those that are actually either specialized on or can feed on, at least, are toads, are shown in dark green and light green respectively. You see them popping up throughout the tree here of snakes. The presence of those amino acids in a given species of snake is shown with a red dot. The absence of those amino acid substitutions is shown with a black dot. So to some extent, you should picture the red dots as being things that can at least to some extent feed on snakes if they were wanting to. Black dots, those that would experience acute toxicity to of the compounds and would not be able to feed on the snakes at all. Now, all these little here these at the tips at our species, these values are known. Everything you're looking at back on the tree, though, are reconstructed, probabilistically reconstructed ancestral character states. The proportion of each of these pie charts, in essence, that's red, is the probability that the trait back then was having the amino acid substitutions, whereas black is the probability of not having them. And you'll see at some places back in time there's high confidence, like all through here, that they had the resistance mutations. Here we're starting to have a little bit less confidence in having them. Here we're very unsure about whether they were there or not. Here it's unlikely that they had the resistance mutations, and at the root of the tree it's less probable for them to have had them than for them to have not had them. Turns out that based on these data, it looks like these amino acid substitutions have evolved at about 10 times, probably independently, and interestingly show up in many lineages that do not in fact feed on toads, which is kind of cool. So ancestral character state reconstruction is a pretty popular thing to do with phylogenetics and can be an interesting way to think about the evolutionary history of characters. But again, can be a little bit problematic if selections had a major role in a character, because then you could get rapid transitions between states if populations or species encounter different environments. The last thing I want to talk about in terms of questions you can get at with phylogenetics includes thinking about variation in rates of diversification through time. The basic question here is, if you think about a group of a clade or a group of species, the question is, have rates of speciation and extinction, which together sort of give you the rate of diversification, right? Think of the rate of diversification as the rate of speciation minus the rate of extinction, the rate at which a group is accumulating species. So have rates of speciation and extinction, and therefore rates of diversification, remained constant through time. And we can sort of think about that. We can visualize that to some extent. So I want to show you first an example of what you might see on a tree if you had constant rates of diversification. You might see something roughly, to the extent that I'm able to draw it, it looks like this. And this will make more sense when we sort of contrast it with the other examples. So where you're basically getting splits at a constant rate. So as you move along the tree, there's a split. You wait a little bit longer, another split, another bit longer, another split. Let's compare that to two different examples of variable rates of diversification and see what the tree would look like. So imagine one possibility would be that there is a period of rapid speciation deep in the past, and there's been a slowdown of speciation since then. That might look something like this, right? Where essentially all of the speciation events occurred in the distant past. And since then, either any new species that have emerged have gone extinct, or the rate of speciation slowed down. So either the rate of speciation slowed down, or the rate of extinction sped up, so that there's been sort of no net diversification since this period in the past. So something's happened to the rate of diversification. You could also get a variable rate of diversification in the other sense of a recent speed up, and that might look something more like, where there was a period when there wasn't a whole bunch going on, again, either because there was a low rate of speciation or a high rate of extinction, then something happened around here in the tree, and suddenly you got these sort of bursts of diversification. Now, quantifying finding evidence of variation in rates of diversification leads to the question of why, right? What happened at this point in history or this point in history to cause a substantial shift in either the rate of speciation or the rate of extinction? So the question then becomes, so if they exist, right? If they exist, are bursts of diversification or extinction associated with important innovations or historical events? In other words, did some key trait evolve in one of these lineages that increased the rate of diversification? Did a niche open up that allowed speciation rate to increase? Here, did niches fill up? Right? Was there some major catastrophe that wiped down a lot of diversity in other lineages and that then allowed this group to sort of speciate more? What's sort of happening there? And there are statistical tests to ask about rates of diversification, and then you could try to tie these to historical events. One example I'll talk a little bit about is evidence of rate variation, and specifically burst of diversification associated with host plant shifts in butterflies. So here, essentially the idea is that by a butterfly group colonizing a new family, for example, of plants, this opens up a new niche for that group, allowing speciation rates to increase or extinction rates to go down, which leads, at least temporarily, to a net increase in rates of diversification. This can be visualized by looking at what's called a lineage through time plot, and I'll show you one of those real quick. So here, you're looking at lineage through time plots for a number of butterfly lineages. I don't really care that you know what any of these are, but each of these panels represents a different group of butterflies. And in each of these cases where the results that are being shown are for a group of butterflies that at some point in the past had a major host switch. Now what you see on the x-axis in each case is time, so going from the past to the present, and on the y-axis is the number of lineages that existed at that point in time. And maybe it would help, I can sort of think about, we can draw that real quick or sort of visualize it. So a tree, going back in time, you can think about how many lineages there were at any point in time. So if you go back to the very root of a tree right here, there was essentially one lineage, right? Now I can look at other parts along the tree. For example, if I go to more recently in time, here there were 2 lineages that my line's crossing, right? At this point in time, there were 3 lineages. Now we have 5 lineages. So as you move to the present, you're going to tend to because of a bifurcating tree, you're going to see more and more lineages, but they should accumulate at different rates, depending on whether you have a constant rate of diversification, a speed up, or a slow down. So back to here, we have plots in green of the number of lineages on a log scale through time for these different groups. And in all of these cases, in a statistical sense, there's significant evidence of rate variation. In some of these, there's an increase in rate associated with host shifts. So this is shown here in this example. And the solid and dotted lines are null expectations from a null model. Here you see a sudden uptick right here, and then things sort of calm back down, then an uptick right here. Both of those correspond with host plant switches in this group. And here, this is in paired butterflies. There was a switch from Fabaceae, so beans, to Brassicaceae, brassica, things like mustards, many of our vegetables we eat. And you see right when that happens, this big jump in diversity, many more lineages all of a sudden, then evens back out. That seems to be associated with that host shift. Let's look at that one phylogeny in a little bit more detail. So here's going back, and you can't read any of the names, I don't care. Here's going back to the sort of ancestor of the paired butterflies. And at this point, D, right here, you get a shift onto Brassicaceae. So think about it from this ancestral species right here. This ancestral species split into 2 things, this one and this one. In the group that stayed on beans, you ended up with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 lineages, right, in the group that stayed on beans. The lineage that hopped on to Brassicaceae instead, I'm not going to count these, but clearly ended up giving rise to way more species, right? There was this giant radiation once you got the shift to Brassicaceae, presumably because it opened up a lot of, again, new niches that allowed the group to radiate and take off, and you had a net increase in speciation rates or a net decrease in extinction rates. So, that's where I want to end with this. Just again, this is hopefully some examples of ways in which phylogenies can be used to address questions in evolutionary biology. For the analytical exercise, I don't necessarily expect you to do anything as complicated as any of these. These are all sort of publishable results. But to think about what you could do that's sort of related to these kinds of questions, right? Perhaps in a simplified framework. Whether you could ask about ancestral character traits, about the relative specios, how speciose different groups are, about historical events, when did something happen, when did something split something else, when there's interest in that context, and in some cases even systematic questions, as long as there's sort of a reason to be interested in that specific question. And that's where I'll end this.